Welcome to our video lecture on neuropharmacology. We're basically looking at medicines, drugs, pharmaceuticals that have an influence on our neurons, especially on our neurotransmitters and synapses. And that's exactly what our essential idea is for today, is that the communication between neurons, again, this is talking about synapses and neurotransmitters, can be altered or changed through the manipulation of the release and the reception of chemical messengers. Those chemical messengers are either hormones or neurotransmitters. So if we change how our neurotransmitters are released, if we change how they bind to protein receptors on the postsynaptic cell, then we can change the communication between our nerve cells. And that's what we're going to look at today. Our objectives are, we're going to, of course, review synapses and neurotransmitters since we're going to talk about them so much today. We're going to compare and contrast both excitatory and inhibitory neurotransmitters. We're going to define this idea of summation, which is literally just like how neurotransmitters add up. We're going to compare and contrast fast and slow neurotransmitters. Sometimes those slow neurotransmitters are also referred to as neuromodulators. We're going to look at lots of examples of psychoactive drugs. We're going to look at stimulants and sedatives. We're going to look at drug action, which can be agonistic or antagonistic. And then we're going to get a little understanding of drug addiction. So first up, a review of synapses. Remember that, if I back up my nerve cell here a little bit, our axons are surrounded by some myelin sheath. That myelin sheath is composed of Schwann cells. This allows for our nerve impulses to be saltatory. They can jump from one node of RNVA to the next. When that happens, we are going to let sodium in. That's depolarization. Potassium gets out. That's repolarization. When we get to the very end of the axon, sometimes we call this the axon terminal or the axon terminus. Sometimes we call it the motor end plate if we're talking about a nerve cell that talks to muscle tissue. Regardless, when the action potential gets to the very end of the axon, another ion channel is going to open up. This time it's calcium. That calcium is going to flood into the cell. It's going to activate these vesicles that are filled with neurotransmitters. It's going to cause them to migrate to the edge of the cell membrane. They're going to dock to the cell membrane. Their membranes will merge. The membrane of the vesicle and the plasma membrane of the axon will merge, because remember that membranes are fluid and flexible. Those neurotransmitters will be released into the synapse. The synapse is that space between the two cells, and then the neurotransmitters can bind to protein receptors that are embedded in the postsynaptic membrane, causing something to happen. Usually we talk about those sodium channels open, and sodium can get in. Here are some of our favorite neurotransmitters. We're going to talk about a lot of these throughout this lecture. Adrenaline and noradrenaline, also referred to as epinephrine and norepinephrine. Acetylcholine, this guy we talk a lot in terms of those neonicotinoid pesticides and how they cause bugs to be paralyzed because those pesticides look like that acetylcholine neurotransmitter. All right, so neurotransmitters can be either excitatory or inhibitory. Excitatory neurotransmitters are the guys that we've been talking about the most so far. They are going to cause depolarization on the postsynaptic membrane. Remember that depolarization is when we have an influx of sodium into the cell because sodium ion channels open. All of that extra positive stuff causes the cell to become positively charged, and that is depolarization. We can also have inhibitory neurotransmitters. They're going to bind to the postsynaptic membrane, but instead of making the sodium rush in, they're actually going to cause often chloride ions to rush in. Chloride is negatively charged. It's going to cause the cell to get more negative. It's basically going to be hyperpolarized instead of depolarized. That extra negative makes it harder to stimulate that nerve cell. And so our inhibitory neurotransmitters basically shut down that postsynaptic cell. Here's that same idea, but in picture form. Our excitatory neurotransmitters on the synapse, something like glutamate, will be released from the presynaptic cell. It's going to diffuse across the synapse, bind to the postsynaptic cell. That's going to cause these sodium channels to open. Sodium gets in, and that is depolarization. We can also have these inhibitory neurotransmitters like GABA is going to be released from the presynaptic cell, diffuse across the synapse, bind to the postsynaptic cell. But instead of sodium getting in, now chloride is going to get in. Chloride is negatively charged, and it's going to cause the cell to become extra negative, hyperpolarized, which is going to basically make it really, really, really hard to get that cell to be stimulated. Summation. Summation is this idea of lots of different neurotransmitters coming together at the same postsynaptic cell and having some sum effect, literally adding together the sum. So summation is literally just adding up all of the different stimuli that are hitting a single neuron. We can have E's and I's. So E is excitatory. The other letters P, S, P stand for postsynaptic potential. So this is an excitatory neurotransmitter, and then an inhibitory neurotransmitter, inhibitory postsynaptic potential. If I have one guy that's like sodium in and the other guy's like chloride in, the positives and the negatives, they literally cancel each other out and nothing happens. We go back to resting potential. So excitatory and inhibitory neurotransmitters are going to cancel each other out. Spatial summation, we have different neurotransmitters being released from different neurons. So they're basically coming from different spaces. So I have one cell releasing excitatory neurotransmitters, another cell also releasing excitatory neurotransmitters. Both of these guys are being received by the same postsynaptic cell, and so I get a little positive from one guy, a little more positive from the other guy, and that's enough to reach threshold potential, and we have depolarization happening. Spatial, our neurotransmitters are coming from different spaces. Temp is all about time, and so now I have the same neuron that's going to send some signals, and then a couple seconds later it's sending more signals, and so the same neurotransmitters are being released from the same neuron, same presynaptic neuron. So we get a little positive from the first boost and then a little bit more positive, and that's finally enough for us to reach threshold, and then again we have depolarization. One neuron sending out neurotransmitters one time is usually not quite enough for us to meet that threshold value. Those are the little sensory things that our brains ignore. For us to send that actionable potential, we've got to reach that threshold value, which usually requires some kind of summation, either spatial from multiple neurons or temporal, one neuron sending more than one signal. Good. All right. So now we're going to look at the idea of fast and slow neurotransmitters. Fast neurotransmitters is what we've been talking about most so far. So these have an almost immediate effect. It takes only milliseconds. A millisecond is 1,000th of a second. So super, super fast, crossing the synapse. These guys are going to bind to the postsynaptic receptors. It's going to cause ion channels to open, sodium or chloride or something gets in. Be careful, fast doesn't necessarily mean a stimulant. It doesn't always mean excitatory. It can. Acetylcholine is an excitatory fast neurotransmitter, but GABA and glycine are inhibitory neurotransmitters that are also fast actors. So we might be sending sodium in really fast. That would be like acetylcholine, but we could also be sending chloride in really fast, which is going to cause the cell to hyperpolarize, and that's what GABA and glycine will do to inhibit cells. We can inhibit things very quickly. Different are our slow neurotransmitters, which sometimes we refer to as neuromodulators. So these are acting in tenths of seconds, which sounds really fast, but that's like 100 times slower than the other guys. So comparatively pretty slow. And instead of just going from one neurotransmitter to the next, they are going to flood onto entire groups of neurons that are surrounding that presynaptic cell. Some examples are epinephrine, norepinephrine, dopamine, serotonin. So these guys are going to bind to a G protein coupled receptor, which is going to cause some stuff to happen. And I have another picture that shows you some more of that stuff. And this guy is going to slide over to another guy, another protein, which in this case is our ion channel. And that ion channel is going to open up, and our ions, either sodium or chloride, can get in. What's super cool is that this guy is going to keep that ion channel open longer, and so the ions can get in for a longer time, which means that that response can last longer. Good? Good. Those slow neurotransmitters, neuromodulators, do not affect ion diffusion directly, but instead they cause the release of that secondary messenger, that G protein, which is going to slide over and then regulate that fast transmission, either open up or shut down those ion channels, which again will last for a longer amount of time. So this fast-acting neurotransmitter bind, open, but then it's going to close right back up. Whereas our slow-acting, we have to wait for this guy to get all the way over there, but then it's either opened or closed for a longer amount of time. We can keep things open for full seconds, which again doesn't sound like very long, but if we're talking about compared to milliseconds, it's pretty long, or even minutes of ions getting into or out of our nerve cells. Here's just one more look at this idea of secondary messengers. So epinephrine, also known as adrenaline, can bind to that protein receptor. It's going to cause this G protein coupled receptor is going to do some stuff, and we're going to flip our GDP into GTP. Just like we have ATP and ADP, we can similarly bind, instead of adenine, we can bind guanine to some phosphate groups, and so that GTP can come and activate another protein embedded in the membrane. In this case, it's adenylate cyclase, because we're talking about adrenaline and anyhow. So we're going to get this guy to turn on, which is going to cause more things to happen. So ATP, remember that's adenine with 1, 2, 3 phosphate groups. If I chop off one, I'm left with 2 phosphate groups, that's ADP. But if I chop off 2 phosphate groups, I'm left with only 1 phosphate group. One is mono AMP. And so we call this cyclic AMP, adenosine monophosphate, and it's going to go turn on more kinases, these proteins that cause all kinds of things to happen inside our cells. So we have a lot of different steps in this process, like this guy has to go over here, and then this guy goes there, and then that guy does that, and then that guy makes those guys do that. It's a lot of steps, but this effect lasts for a very, very, very long time. And so it is going to be an important way for us to regulate our nervous system. And the coolest thing that it can do is it's actually going to change the number of protein receptors on the postsynaptic membrane. So here is maybe before we had that slow-acting neurotransmitter, I've got 1, 2, 3 proteins embedded in the postsynaptic cell that can bind to the neurotransmitters that are being released. After I've got these slow neurotransmitters causing this secondary messenger effect in here, now instead of only 3, I've got 1, 2, 3, 4, 5 neurotransmitter protein receptors available to grab onto those neurotransmitters and do all kinds of cool things. So we're going to change the number of receptors with our slow neurotransmitters. That's going to change the rate of ion diffusion. I can get more sodium channels to open. I can get more chloride channels to open because I've got more receptors that can grab onto those neurotransmitters. This is super important for learning and memory and brain plasticities when we talk about connecting the brain synapses, neurons in different ways, training our brain to do different things with different parts. We think that these slow neurotransmitters are super important for the remodeling of our synaptic connections, growing new synapses, redirecting our synapses. And our secondary messengers can have an effect for days, so seconds, minutes, days, and that's going to lead to what we call long-term potentiation. And you can see before our long-term potentiation, after our long-term potentiation. We've got a much, much bigger response, and so we can send those signals faster, more easily, and this is the result of our learning, practicing, stabilizing those synapses. All right, so let's start talking about some of the medicines and drugs that can impact how our neurotransmitters, fast and slow, can work. So psychoactive drugs are drugs that basically are active on our neurotransmitters, on brain and neurons. They're going to either increase or decrease neurotransmitter movement, that neurotransmission. Our excitatory drugs are going to increase neurotransmitter movement. Some examples are nicotine, MDMA, and amphetamines. Inhibitory drugs are going to decrease transmission of our neurotransmitters. They're going to calm us down. Some examples are benzodiazepine, valium is an example, alcohol, and then THC, which is in marijuana. IB might ask you to state examples of excitatory drugs and inhibitory drugs. Luckily, BioNinja to the rescue with another amazing mnemonic device, Batman. If you need to name some drugs, just remember Batman. So our sedatives or our inhibitory drugs, BAT, benzodiazepines, valium is an example, alcohol, THC, which is in marijuana, and then our stimulants or our excitatory psychoactive drugs are MAN, MDMA, amphetamines, and nicotine. Just remember Batman. All right, so now we're going to look at drug action, so how drugs actually work. So here is like before a drug is introduced to synapses. We have our natural chemical. This is basically the neurotransmitter that is supposed to be doing this job, and it's going to bind to its protein receptor, and then stuff happens. Normal cellular activity, sodium channels open or chloride channels open, something happens. An agonistic drug is going to mimic that neurotransmitter. It's going to bind to the receptor site just like the neurotransmitter would, and it's going to do the job of the neurotransmitter. We're going to enhance that activity of the neurotransmitter. Antagonistic drugs are opposite. They're actually going to block the receptor site so that the neurotransmitters cannot do anything, and we have blocked activity. So agonists are going to mimic our neurotransmitters. Antagonists are going to block our neurotransmitters. Anesthetics are painkillers, and it might be natural to assume that they are blockers, that they are antagonists, but in fact, they work in an agonistic way. So most of our anesthetics are agonists of endorphins. Endorphins are natural compounds that our body makes when we're super happy, and they block pain. How they do that is this. Our endorphins are released from a cell. They are going to float across some synapse. They're going to bind to some proteins that are known as opiate receptors. So our endorphins are going to bind to opiate receptors. This binding is going to prevent these pain signaling neurotransmitters from being released. So the pain signal never gets to the brain. So we don't know that we're hurt because endorphins are going to stop the pain from the pain signals from being sent. So painkillers are going to work the same way. So we can add some more of these guys, and they're going to bind to those opiate receptors, which is going to block those pain neurotransmitters from being released, and then our brains don't know that we hurt. So even though they're blocking the pain, they're actually increasing the activity of the endorphins by binding to the opiate receptors, just like endorphins would. Stimulants, we'll look at stimulants for a little bit. They're going to mimic the sympathetic nervous system. This is our fight or flight. They're going to increase heart rate, increase temperature, increase blood pressure, make everything go faster. We're more alert, we're more energetic, we're more confident, interestingly. Some examples, caffeine in coffee, theobromine, which is in chocolate and cocoa, are considered legal stimulants, and then some less good choices for stimulants are cocaine, amphetamines, nicotine, which is in cigarettes and cigars, chewing tobacco, and then MDMA or ecstasy. These are all stimulants. Remember, this is the man part of Batman. MDMA, amphetamines, nicotine. Good. So here is an example of a stimulant, oh my gosh, I can never say this. Pramipexole. Pramipexole is an agonist. It's going to mimic the action of dopamine. So we can add this drug as a stimulant. It's going to act a lot like dopamine. And it's used to treat depression. It's also used to treat Parkinson's. And so Pramipexole, or there's another example called L-Dopa, which is literally like levodopa or a dopamine mimicker. It's going to work just like dopamine would. It's going to basically cause the dopamine receptors to bind more often, and then we have that happy feeling of dopamine. It is an agonist. Cocaine is also a stimulant, but it is an antagonist. So that dopamine is going to be released. Voila, it binds. And then what should happen is that dopamine gets sucked back up into the cell, recycled and used again. What cocaine does is it blocks that uptake of the dopamine. So we should have dopamine bind, feel happy, dopamine goes away. What's going to happen with cocaine is that dopamine is released and it stays there and it stays there and it stays there, and there's so much happiness that you go a little crazy. I'm making it sound like you're so happy. Maybe you know cocaine is good. It's not good. Don't do it. We like normal amounts of dopamine. This is bad. But it is an antagonist as opposed to the last drug, which was an agonist. So cocaine blocks the uptake of dopamine, which means that dopamine sits in the synapse longer, increasing the effect of the dopamine. Diazepam or valium is an agonist. That is a sedative. It is going to act just like GABA. GABA is one of our neurotransmitters that causes chloride channels to open, which makes the cell hyperpolarized. Remember, depolarization, we get more positive. Hyperpolarization, we get more negative. This basically turns the signaling in that nerve cell off. So diazepam is going to act just like GABA. It binds to GABA receptors. It's going to open up that chloride. It's going to shut down the cells. And when it shuts down the cells, it reduces the feelings of anxiety. It reduces panic attacks. And it also reduces insomnia, helps some people sleep. It is an agonist. It acts like GABA. THC in marijuana is an antagonist. So it is actually going to block the release of excitatory neurotransmitters. So it is going to bind to what we call the cannabinoid receptor. It's going to prevent those excitatory neurotransmitters like acetylcholine from being released. And so there's just less neurotransmitter being received on that postsynaptic cell. And so you just feel more quiet. All right, neonicotinoids, remember, just a little bit of review. Neonicotinoid pesticides are agonists. They act a lot like acetylcholine. They are going to bind to the acetylcholine receptors on the postsynaptic neuron. Usually we have the super cool enzyme acetylcholinesterase that's going to break down the acetylcholine so that it can get taken up again and used again. But the neonicotinoid pesticides cannot be broken down by the enzyme. The binding is permanent. No more acetylcholine can be, well, the acetylcholine can be released, but it cannot bind to the receptors, which means that this cell can't be signaled, and so the insects that eat this pesticide are going to be paralyzed. All right, quick slide on drug addiction, and then, my friends, we are calling this lecture done. So drug addiction is a chronically relapsing disorder. So some of the signs of drug addiction, it does have to be chronic, it has to be over time. There is a compulsion to find the drug and then take the drug. You cannot limit, you cannot control how much of the drug that you take, and then you feel a very negative emotional state when access to that drug is prevented. Some of the risk factors for drug addiction, some of us tragically have a genetic predisposition. If you have parents or grandparents who were addicted to alcohol or drugs, you are more likely to be an addict. You're not necessarily for sure going to be, but your risk is higher. Environment, if you are around a lot of people who are taking drugs or drinking alcohol all the time, you are more likely to become addicted to those substances. And then your dopamine levels are also going to lead to drug addiction. So drugs that affect your dopamine levels are most addictive. So opiates and cocaine, nicotine, and alcohol that all impact dopamine are going to be the most addictive of the drugs. Don't do them. They're bad. All right. Hopefully we feel like we have met our objectives for the day. We reviewed synapses and neurotransmitters. We compared and contrasted excitatory and inhibitory neurotransmitters. We talked about summation. We looked at fast and slow neurotransmitters. Those slow neurotransmitters are sometimes also called neuromodulators. And instead of opening ion channels, they actually change the number of neurotransmitter receptors on those postsynaptic cells. We looked at some examples of psychoactive drugs. We're going to think about Batman, our sedatives, our benzodiazepine, alcohol, and THC, which is in marijuana, and then our stimulants are man, we have MDMA, and amphetamines and nicotine. They can be either agonists, where they mimic neurotransmitters, or they can be antagonists, where they block those neurotransmitters. And then we talked a little bit, just that one little slide, about drug addiction and how dopamine plays such a big part in addiction. Good work today, my friends. I will see you in class.